There are more than 40 neurodegenerative disorders caused by DNA repeat expansion, such as ALS and Huntington's disease. Out of them, more than 24 are caused by trinucleotide repeat sequences. Once the number of repeats passes their threshold length, they become uncontrolled and produce toxic dipeptide proteins which cause neuromuscular degeneration. Current detection methods rely on PCR methods, which are prone to frequent false positive and false negative results. So our research is geared toward developing simple and label-free methods to detect the associated repeat expansions, such as a CAG repeat for Huntington's disease and the CGG repeat for Fragile X syndrome. Repeat sequences have sequence-dependent unique structural and biophysical properties, and our lab focuses on using those biophysical properties to detect the expanded repeats by amplification-free and label-free methods. We have 3 simple methods to detect the expanded repeats, which I would like to discuss here. The first project uses wax-on plastic microfluidic channels printed on PET substrate to detect the Huntington's disease-causing repeat CAG. So this study uses the sequence-dependent flexibility of the repeat sequences, which can be translated into a change in the capillary flow in the microfluidic channels. Flexibility is affected by sequence type and length of these sequences, as shown in these 2 charts here. In the first chart, we can observe the difference in flow velocity based on the varying flexibility of the different types of repeats. This is just based on some properties of the bases in the strands. Here you can see that the sequence GAA, it has the least flexibility because it's showing the highest flow velocity. In this chart here, you can see that the length also impacts flow velocity. The higher the length, the lower the flow velocity. So you can see that the highest number of repeats here of the longest length is exhibiting the lowest flow velocity. Our wax-on plastic devices were able to detect a difference in capillary flow between patient-derived normal and abnormal CAG repeats associated with Huntington's disease, where we can see here in this figure that the Huntington's disease positive repeats have a significantly lower flow velocity than the normal number of repeats. And this assay is completed within 2 minutes, and the sample volume required is less than a microliter. The second project here demonstrates that the absorption of Fragile X syndrome causing CGG repeats with 2-dimensional MOS2 nanosheets enhances the interfacial electrochemical current, which enhances with the increase in length of the repeats. So here you can see in this chart that as we increase the length of the repeats, we're also seeing an increase in current. Even just a difference of 10 base numbers from 20 to 30, we're seeing a very significant increase. So even here you can see like this 100 number of repeats and the disease causing number of repeats is showing a significantly higher current than the normal number of repeats here. And we monitored the current of these repeats on our wax-on plastic electrochemical devices modified with MOS2 nanosheets. And the limit of detection of this platform is down to 3 attomolar. Our third project here involves surface-bound PNA microprobes immobilized on a gold surface, which captures the CAG repeats that cause Huntington's disease, resulting in a change in the charge transfer resistance of the biosensing platform. The change in resistance is measured in the presence of high concentrations of the magnesium ion, Mg2+, using label-free electrochemical impedance spectroscopy technique. The magnesium ion neutralizes the negative charge of the DNA targets, and surface impedance is only the function of the length of the target. This increases the resistance with the length, and the change in resistance before and after hybridization of the target was used to compare abnormal and normal repeats in total RNA extracted from neural stem cells. So as you can see here, as the length of our repeats increase, you're seeing an increase, a proportional increase in resistance. And that charge transfer resistance that we're measuring is just the diameter of the semicircle here that we're seeing. And here you can see in red is the disease-causing number of repeats, and this is in real RNA sample. And green is the normal number, so you can see a significant increase in resistance there between the normal and disease-causing number of repeats. And this method actually has a 100-fold better sensitivity than we reported using DNA microprobes than in our past study. So overall, our lab has developed various strategies and devices with a wide range of potential application in the diagnostics of genetic disorders caused by trinucleotide expansion repeats. Wax-on plastic sensors are low-cost, flexible, and versatile platforms we have developed in our lab for a variety of applications. And you can find relevant publications of our research on our website at mshampsy.com. Thank you.